Okay, so let's continue with this chapter about memory. So in this figure we can see a maze, a radial arm maze. Usually what happens here is that if you put a rat in the maze and the maze has many arms with food at the end of each arm, the rat will explore each arm, they will eat the food, and then they won't go back into the same arm twice because they already remember that they ate the food at the end of that arm. So this happens with rats who has very good long-term memory, which means they have a preserved hippocampus. If you perform surgery on rats and you remove the hippocampus, then you will find that rats perform similar to normal rats, meaning they go to the arms and they explore them and find food, but usually they go back more than once to the same arm, and sometimes they even miss some arms with food, meaning that they do not remember, they have some problems to remember the places they've been before, and this is directly associated to the hippocampus lesion. And this is an experimental design made by Morris, and it is a pool consisted of a pool with water, a milky water with color, white color, so the rats cannot see what's under the water. And this is used to test spatial memory. So rats usually swim through the pool until they find a platform in where they can stand and breathe. And after you put the rats in this pool several times, they will find the way directly to the platform, meaning that they develop some sort of long-term memory for place, for space. And this figure is showing actually action potentials fired for what they call place cells. So very similar to what we studied before. I don't know if you remember, we were talking about receptive fields of neurons, some neurons that respond when stimulus are presented in one specific place on the screen. So in this case, neurons respond when the rat is standing in one place in a maze, and usually one particular neuron fires for one specific place, and so they call these neurons place cells, and they were found in the hippocampus. So this image is showing red colors mean more action potential and blue color less action potentials. They're showing two kinds of cells which have specific, the rats move all through that rectangle, but the neuron only fires when the rat is exactly standing at that point. That's why they call them place cells. This is an experiment related to spatial navigation in humans, and you can see also that there is increased activity in the right hippocampus and caudate nucleus. The caudate nucleus is one of the basal ganglia, and also part of what we call a striatum. And this again place cells, and you have two different patterns. One pattern in figure A, the cell responds when the rat is in one region of the enclosure, and B is a grid cell in the entorhinal cortex, very close to the hippocampus, and it is active when the rat is at multiple locations in that form of a grid pattern. Also, the red colors mean action potential, so the cell is firing in those specific places of the maze. This is another experiment they did using different stimulus, in this case odors. So the rats remember places by their smell memory, memory for smell, different smells. And this is a very good image. This is showing actual theories about how memory are created and storage and retrieved. Now what you can see here is usually the main idea, the main theory or hypothesis that scientists have about this is everything we perceive is have to pass through hippocampal neurons in order to be stored again in the cortex. So you can see here that a new memory is created, and there is a strong relation between hippocampal neurons and the neocortex. And usually the idea is that after that memories are storage in the neocortex. So you can see in figure B strong lines, the black lines, continuous lines mean the memory, and you can see in dotted lines would be the previous pathway they follow to be created. And the figure C is showing what might happen during retrieval. When you remember something again, that hippocampal neurons are activated, and they activate the memory which is storage in the neocortex. And this will be the green color. Red color may be showing some other memory associated to that main memory. For example, when you remember something, you might also remember the smells or some sounds related to that specific episode. Those are similar memories that are storage created at the same time, and they usually retrieval together with the memory you want to remember. This is an experiment showing the relation between some other brain areas different from the hippocampus in memory. In this case, the striatum. Striatum is the caudate nucleus. You have to remember the basal ganglia. There are usually three nucleus in the midbrain. Not in the midbrain, but in the base of the brain, let's say. They are the putamen, globus pallidus, putamen, and caudate nucleus. So putamen and caudate nucleus are called together the striatum. And they show here the relation between the response of this striatum cells also in some memory task. And the same happens here, but this is with human patients. So in this case you have two profiles, some patients with amnesia and some patients with Parkinson's disease. And we know Parkinson's disease is related to some impairment in the striatum. I mean the connection between midbrain and the striatum. So you can see that the performance of people with Parkinson's is worse when you have to perform a task that requires what we call procedural memory. Is what is shown in figure B. So they have a problem to remember, for example in this case, the order in which these cards are presented in order to predict the weather. But if you take another test, for example questionnaire, the normal typical test, the Parkinson's patients performed almost exactly like normal patients, and amnesia patients performed really bad. You can see that even though there is a relation between striatum and memory, it is more related to procedural memory rather than declarative memory. And that will be all for this chapter, so we will see in the next chapter. Remember to answer the questions and handle it to the classroom. See you.